Hello everyone. So I'm getting on here real quick to explain what's going on with the algae hormone experiment. If you saw my video yesterday about the day one results, you'll see that my indole acetic acid treated algae essentially exploded. They essentially underwent apoptosis. And essentially nothing has changed. The entire culture has essentially died. I've decided instead to redesign my experiment. And the reason for this is a little backstory when it comes to the literature, when it comes to hormones administered to algae. Now I'm dealing with spirulina algae, and there isn't much literature out there. There's, I believe, 4 studies altogether that I can find that do any sort of research on administering indole acetic acid to algae. Now there are many studies done on other types of cyanobacteria in algae, and the one thing that I've noticed with the papers is so not none of them really do any sort of cell count measurements. It's usually absorbance. So essentially, you pass a certain wavelength of light through, and a certain percentage of light gets absorbed, and then you can use that to calculate the density, the cell density, which you could theoretically you can make a standard curve, and then you can kind of estimate the amount of algae present in a sample. None of them actually do that. They don't actually do that. The absorbance is enough. Now, some of them don't even do absorbance. Some of them do dry weight of the algae as a way to calculate, and a bunch of other different ways. But essentially, they don't get down to the actual amount of cells that are per milliliter in the sample. Also, I think only 3 or 4, if I remember, of the studies that I read, could you actually calculate or at least know the volume of the sample to begin with, and none of them, there's no way to actually know how many cells per milliliter there were at the beginning of the experiment. With this in mind, if you look at the data, even among similar types of cyanobacteria that have been administered indole acetic acid or other types of hormones for that matter, one thing you'll see is that with all of them, they kind of have the same sort of data, the same curve, I would say. As you administer it, the amount of growth increases to a point, and then almost immediately it drops off. And that means there's a toxicity level in which there's no way they can grow beyond a certain concentration, right? That number is different in each study. Even among the same cyanobacteria, the concentration level goes anywhere from 10 to the negative 8 moles concentration, all the way up to -4 moles concentration. And my thought is that there's no real, as of yet, the literature does not show any, there's no way to know how much hormone to administer to a given sample. So what I'm doing is I'm going to redesign this experiment to where I have the same concentration, which will be 10 to the -6 moles of indole acetic acid, but I'm going to vary the amount of cells in each sample. And my thought with this is there's a certain amount of receptors. Essentially, each trichome of algae can only handle a certain amount. And my thought is it's not the concentration that matters, it's the amount of algae that's present. Because if there's enough algae to consume a certain amount of hormones so that none of the algae ever exceed their toxic level, then you'll have a growth. You'll essentially have stimulated growth. But if the amount of algae is below a certain concentration, they will just keep absorbing the hormone until they've essentially been overstimulated to the point where they blow up, like they've been put in a blender or something. And so what I want to do is I kind of want to turn the experiment on its head, and instead of doing a concentration of hormone experiment, I'm going to do a concentration of algae experiment. And what I want to see is at what point, my thought, my prediction is that below a certain level of concentration of algae, the algae will completely collapse. Like all the cells will die, just like what happened in the incubator. But then once you get beyond a certain point, you'll start to see there's a decline, but the decline will be of a certain level. Like, let's say you start out with 1 million cells, right? You might have completely dead, and then you reach a point where you might have 100,000 cells, 200,000 cells, 300,000 cells that survived out of the million. And then eventually you'll get to the point where it'll be 1 million. Essentially the effects of the hormone will be balanced out. And then beyond that point, you might get, let's say, some increase where it grew, and then it actually reaches a certain point where it's almost like there's too many cells and they didn't consume enough, and then it levels off again. And that's my thought with this is I would do, let's say, 20 different concentrations of cells. So essentially 20 control and 20 experimental. And then each with a pair that would have, let's say, 100,000 cells, 200,000 cells, and so on. That way I can track the difference in growth rates of all the different samples. And my thought is that I would administer the same concentration of hormones to all of them, and then put them in the incubator for maybe a week. Let's say I'm still working on exactly how I'm going to do this experiment. Because of the volume of it, I can't exactly do huge jars. They're going to have to be small test tubes. Like I can't put them in there for more than a certain amount of time, because eventually all of them will start running out of aeration and carbon dioxide and all that kind of stuff. And make sure to check, subscribe, and do all that kind of stuff, and hit the notification bell if you want to follow this variation of the experiment that I'm doing. Thank you.